Start. Okay, and it's a recording in progress, so.
I'll turn my camera off and audio off.
Okay. Hello, everyone logging in. We'll just give it a few minutes here for folks to join. Okay, it's now 1 o'clock, but we'll still give it maybe one more minute for a few folks to join, and then we'll start the webinar. Okay, there's still a few folks rolling in, but I think we can get going because we have a very fascinating presentation today, and I think having the full session will be important. So if more folks are joining, welcome. Hello, everyone. Welcome back to a new season of the Ask an Expert webinar series for 2025. My name is Meg Mills, and I'm the International Watershed Coordinator with the Lake of the Woods Water Sustainability Foundation. I also work part-time with the International Joint Commission, collaborating with many partners across the rainy Lake of the Woods Basin. I'll be your host for today's webinar and for the rest of this series as well. Today, I'm also supported by Todd Sellers, the previous ED of the Foundation, who will assist with moderating the webinar during the Q&A and discussion session. I'm joining you today from Treaty 1 territory where it's damp and rainy, but still a lovely day to learn something new. So today we're joined by Dr. Vince Pallas, Head Research Scientist at the IISD-Experimental Lakes Area. Vince and his team have been conducting groundbreaking experiments to better understand how diluted bitumen or dilbit affects freshwater ecosystems. For anyone unfamiliar, Dilbit is a form of heavy petroleum product made by mixing a solvent with bitumen, a thick, tarry type of crude oil. This process makes it thin enough to transport by pipeline or rail, but it's highly toxic to aquatic organisms and poses real risks to our waterways when spills occur. Vince's research explores what happens when dilbit enters freshwater environments, how it behaves and settles, and how ecosystems respond to cleanup efforts. Before we dive in, I'll just go over a few quick housekeeping points. Okay, so this session is being recorded. You'll be able to rewatch it later at our website, along with all of our past sessions and future sessions. If you're new to Ask an Expert, welcome. This series highlights the incredible science and stewardship happening across the rainy Lake of the Woods Basin and beyond. Most of you are familiar with Zoom, I assume, but just as a reminder, please keep yourself muted throughout the session. Actually, in fact, I think you'll all be forcefully muted. You can access controls at the bottom of your screen to adjust your audio or use chat and Q&A features. If you're using the mobile app, you can access Zoom controls by tapping anywhere on the active screen. A taskbar should pop up at the bottom of your screen where you can quickly mute yourself and shut off your video feed by swiping right. You can also restore your sound and video by swiping left. Feel free to type your questions into the chat Q&A box at any time. We'll be keeping an eye on these and we'll save time at the end for questions and discussion. You can access the chat window through the bottom menu, and you can type questions or comments in the chat at any time. Just remember that everyone will see your chat. Even if you do it just to one participant, I'll be able to see the chat log at the end. So just briefly about our speaker today, Vince is an aquatic toxicologist with over 25 years of experience assessing and mitigating the impacts of contaminants of freshwater ecosystems. He has worked extensively with industry, governments, and communities across Canada on issues ranging from agriculture and hydroelectric development to mining and oil and gas. He also has pardon me. Vince also has expertise with environmental effects monitoring under Canada's regulatory framework and brings that knowledge into his role at IISD-ELA. In addition to his research, Vince is an adjunct professor at the University of Manitoba, teaching in the departments of biological sciences and environment and geography. His experience has been sought by organizations such as UN, Environment Canada, Health Canada, Natural Resources Canada, and the US EPA. Just briefly, a quick overview of what we'll be covering in this session. We'll be understanding diluted bitumen, what it is, and why it's such a unique challenge for freshwater systems. Different experiments at the IISD-ELA, how Vince and his team design real-world studies to examine what happens when dilbit enters lakes. We'll also talk about ecosystem impacts and recovery, what the research tells us about how aquatic life and water quality respond over time, response and remediation innovations, and implications for freshwater management, what this means for all of us working in water governance and spilled preparedness. After Vince's presentation, we'll open the floor for a question and answer and discussion. So with that, it's my pleasure to hand things off to Vince. I'll exit this, and you should be able to share your screen, Vince.
Okay, just let me know when that slide comes up. Thanks very much for that introduction, Megan.
Okay.
Is that slide up now?
It's not up yet.
Okay, let me try again. How about now?
Yes, it's just loading. Okay, it's up now.
Great. Yeah, thank you very much for the invitation to speak today. I'm really looking forward to this. This is something we've been working on for many years at the Experimental Lakes Area, and it took quite a few years to get together. And it took us about 6 years to conduct the studies, and in fact, we're still following some of the recovery of those systems, as Megan suggested. So before I start, I just want to a couple land acknowledgements. The IISD Experimental Lakes offices are in Winnipeg. They're on Treaty 1 territory. But our field station in northwestern Ontario, where most of this work occurred, is on Treaty 3 land and the homeland of the Métis Nation. And a large part of the project that I'll talk to you about today had a great deal to do with our regard to these treaties and moving forward in partnership with Indigenous communities. So one of the reasons it took us a while to assemble these studies is we wanted to have a number of different partners at the table. So that included industry, regulators, local communities, so that there was an understanding what we were trying to do before we went ahead and did it, right? Because there's always and I'll mention this several times through my talk, we always have to be able to return our lakes to their original condition. That's the proviso by which we're allowed to do some of the work that we do at ELA. Now, I think most of you know where the Experimental Lakes Area is located, but for those of you who don't, and what it is, it's a set of 58 lakes that are set aside for research in northwestern Ontario. We're located about 60 kilometers east of Kenora. If you head down the Trans-Canada, about 60 kilometers, then make a sharp right, about 30 kilometers down a gravel road is where our field station is. So there's no cottages, there's very little human influence in this area, and so we really are working on very pristine lakes. The field station itself includes labs, accommodations, support buildings, and it was formerly a government-run facility, but now it's operated by the International Institute for Sustainable Development, which is an international think tank, and that's focused on environmental research in favor of protecting Earth's resources and specifically protecting aquatic resources. So for more than 50 years, since 1969, we've been studying the lakes, the 58 lakes that I spoke about earlier, to understand how those ecosystems can be impacted by influenced by humans. So this is a list of brief mission statements that I won't read, but I do want to point out the top one. So while we examine natural processes to understand things like climate change, we also have special legislation, and I've shown those at the right-hand side here, that apply to these 58 lakes that allow us to manipulate those lakes. So over the past, our work has had a great influence on environmental policy related to things like algal blooms, acid rain, mercury, and more recently on pharmaceuticals. And if you want to read more about that work, please take out your phones and click on that QR code link in the bottom right-hand corner. So the work on oil spills that I want to talk about today was also heavily influenced by the last two boxes here of our mandate. And that is examining innovative approaches and communicating with multiple stakeholders. And it came about, like most of the studies at ELA, because of some important data gaps on a topic of public and scientific interest at the time. So planning for the whole ecosystem studies, like I said, is a long-term process. And it really began just as I was rejoining the Experimental Lakes Area back in 2016. So I did my graduate work at the Experimental Lakes Area when water was first invented back in 1987. At the time, around 2016, there was a lot of social and political heat around the production and transportation of oil. So that included the proposed Northern Gateway pipeline, and that was supposed to be one 36-inch diameter pipeline carrying about 500,000 barrels of diluted bitumen from Fort McMurray to the Kitimat region of BC. But as Megan mentioned at the beginning of the talk, bitumen is too thick to flow through a pipe. So you really need to dilute it with something, and we don't have enough diluent. And so the Northern Gateway was actually two pipelines. The second one was a smaller one that would carry 200,000 barrels of condensate the other way that would be mixed with diluted bitumen and then shipped to the Kitimat region. So that's a lot of shipping of oil products. So the same year that that pipeline was proposed, 2016, there was a rupture of a Husky oil pipeline that released about 225,000 liters of blended heavy crude oil into the Lower Saskatchewan River near Maidstone, Saskatchewan. So the cleanup for that spill exceeded around $100 million. Add to that, that 2016 was just a year after the Paris Agreement was signed, and there was a lot of social pressure mounting to make explicit commitments to reducing fossil fuels. And this led to global demonstrations collectively referred to as the Break Free protests. And you could say that, gas and oil were under fire. There were also some important scientific data gaps regarding the impacts of oil at the time. So in 2015, this book or report that I'm about to talk about really became the Bible for how we would conduct the studies at ELA. This was a 2015 report by the Royal Society of Canada. So this is a 500-page compendium on what was known about crude oil spills. And it also identified important data gaps, and it was put together by an independent review or independent panel of scientists. So it outlined a couple of data gaps. First of all, that baseline data at a spill is often lacking. So you never know where oil is going to spill and when it's going to spill. So understanding that system before oil gets into that system oftentimes is just not known. There's also a need to assess the productivity and the trophic interactions. In other words, how ecosystem level effects come to be. And you can't do that in a laboratory. Sorry about that. My slide is auto advancing here. You can't do that in a laboratory. You really need to do it at a whole ecosystem level. And then we also need to know something about the resiliency of aquatic systems. How do they recover? And how do we best remediate an oil spill? So one final word before I delve into the experimental work. I work for IISD, and many of my colleagues in our Geneva offices are vehemently opposed to us working on oil. And the reason is they believe that oil is being phased out. Well, we live in Canada, and energy and oil are closely tied and will be for some time, despite our best efforts and best interests in going towards greener energy production. So the production and exports of oil have been steadily rising since 2010. And currently more than 37% of our energy comes from oil. And that's almost 2 million barrels a day. So even if we accept net zero projections, by 2040, Canada will still derive almost 25% of its energy from oil. So if we're going to be using oil, we better know how to clean it up if it spills, because that's an eventuality, despite how safety records have improved over the years with transporting oil. So the oil spill studies that we conducted can more accurately be described as a program. And it included 3 major projects with quite different offshoots from each of those. And each of them had different objectives. So the first one I've shown here is the Boreal project. That's the Boreal Lake Oil Release Experiments by addition to Limner Corrals. Admittedly, a little bit of an awkward acronym. It examined how oil changes and both from a chemical perspective, but also from a physical perspective as it's spilled onto water. And it also examined how oil accumulates and affects aquatic organisms. The next part of the program was called the Freshwater Oil Spill Remediation Study, or the FOREST project. And it focused on how best to clean up oil after it's spilled on water and then becomes redeposited on the shoreline. And then the final study that I won't talk about too much today is the floater study, and that's the floating wetland treatments to enhance remediation. This is using plants to actually speed up the recovery from oil spills. I won't, as I mentioned, talk too much about the floater study today, but I'll concentrate on the first 2 studies and then give you an idea of what we did in each one of those studies. So beginning with the Boreal study. Boreal study was conducted from 2017 to 2019 and included a year of baseline data collection in 2017. The experimental additions happened in 2018, and then there was a cleanup phase in 2019. So the Boreal project actually couldn't have been carried out without the expertise and funding provided by Environment and Climate Change Canada and NSERC, and an NSERC grant awarded to Jules Blaise from the University of Ottawa, Mark Hansen from the University of Manitoba, and Diane Orahel from Queen's University. I should mention that Jose Rodriguez-Gil, who is now a scientist with ELA, was a postdoc at the time and was responsible for overseeing just about everything that I'm going to show you here today in terms of conducting that field experiment. So it's important also to note what I'm going to show you is the final study in Lake, but there were a lot of preliminary studies that occurred before that. We always sort of proceed in a stepwise fashion, going from, small bench scales to small mesocosms, and then finally, before we get into a lake, you only get one chance to do some of these studies in the lake, and so you have to make the best use of them. So boreal concentrated, again, as Megan suggested, on the product produced by Canada's oil sands region, and that's specifically bitumen. And most of you know, as you can see from the picture here, bitumen is very thick, viscous product that must be diluted to allow it to flow through a pipeline before it can be distributed. And it comes from a couple of different deposits up near Fort McMurray, Alberta. So what we typically hear about is called dilbit, and that's simply bitumen, the heavy viscous stuff, the oil sands, that's combined with diluent, again, to allow it to flow through a pipeline. The ratio of the diluent to the bitumen varies by season and by the mode of transport. But generally, if you're talking about pumping it through a pipeline, it's about 30% diluent or natural gas condensates and 70% bitumen. So the two components differ in their chemical profiles. They're quite different, actually, but also in their fate and effects. So the diluent is highly volatile. So I've shown a couple of the chemical structures here. We're talking about things like BTEX is an acronym that stands for benzene, toluene, ethylbenzene, and xylenes, which is a group of one-ring compounds. Each one of these comprise the entire BTEX. And then they're small, and I'm going to talk a lot about these kind of things. These are called polycyclic aromatic compounds, and I hope you can see my pointer here. So polycyclic means there's a couple of cyclic, the ring structure, and poly, there's a couple of them. This is the smallest known polycyclic aromatic compound. It's called naphthalene. The third portion of diluent are these short-chained alkanes. So each one of these components in the diluent is pretty easily removed from an oil spill site. And that's because it's so volatile, they actually evaporate from that site. But the PACs, and particularly some of the larger ones in the other component, and that's the bitumen, and you can see some of these larger PACs, some extremely large ones, are very persistent and very difficult to break down. When they do break down, oftentimes they break down into 2 or 3 or 4 or 5 ring structures. And those are the ones, despite the fact that they only comprise 1% of the oil, those are the ones that impart the greatest toxicity to aquatic organisms. Now, the density of bitumen, a diluted bitumen, is very close to water. And at the time we were conducting these studies, there was some debate as to whether or not Dilbit would sink when it found its way onto water, if there was a spill on water. It seems like a very basic question, doesn't it? Will the oil sink or will it float? Well, again, it changes over time as this diluent evaporates off. And so that question was key to one of the key issues for the boreal study. So I mentioned before, ELA has special legislation that allows us to be able to manipulate lakes. But again, that permission comes with the proviso that we return those lakes to the original condition. So actually conducting a whole lake oil spill study was not practical or ethical, right? We couldn't assure Ontario that we would return it to its original condition. And so we used an approach that we used many, many times, and that's something called a limnocorral. And I've shown a schematic of it here. These are basically just large enclosures, again, called limnocorral. And it consists of a yellow flotation collar here that sits on the surface of the water. And that suspends a curtain that goes all the way down to the surface of the lake. And we seal it to the bottom of the lake using a double row of sandbags. So essentially what you've done is isolate a column of water and the underlying sediments so that you can manipulate that as a model for the entire ecosystem. Now, these ones were particularly large. These are 10 meters across, so almost, a little bit more than 30 feet across. And you can see some of the other paraphernalia that was installed to allow us to be able to sample inside that enclosure without disturbing it, right? So the oil slick is sitting on the surface here. Anything that went in or came out would be coated in oil. And we wanted to analyze the water independently. So you see some ports here for sampling water. You see an oil-free region here because of this partition. And then we also put sediment jars into the bottom of this enclosure so that we could see over time how much oil accumulated in the sediment. So to make sure that we're working at environmentally relevant concentrations, what we did was to review historic and likely future oil spills, if there ever were, to use a gradient design. So we didn't just want to use one concentration of oil on the water inside these enclosures. Again, Jose Rodriguez reviewed all that data over the past decade and came up with a gradient design that reflected concentrations of oil that had been spilled onto water from small spills to large spills. Again, we want the comparability of these studies to be to its greatest extent. So the model showed us that we should add between 1.6 and 160 liters of diluted bitumen to each of 7 different limnocorrals, and we did that in June 2018. So that's equivalent to a ratio of oil to water ranging from 1 to 100,000 to 1 to 1,000, again, reflecting sort of spills from the past. But it was a very warm day, the day we added oil. And so actually, our target amounts were a little bit more. So instead of 160 liters, it was more like 180 liters. And so the actual oil to water ratios that were encompassed ranged from the 75th percentile to the maximum oil to water ratio that was encountered for a spill in North America. And that was a Kalamazoo River spill in 2010. And that had an oil to water ratio of around 1 to 500. And if you're interested in the Kalamazoo River spill, I mean, just Google that, you'll come up with a lot of websites to read about that. So included in this, we also had near field and far field controls, because when you put up an enclosure, you can affect the ecosystem itself just by virtue of having an enclosure. So I'll use my pointer again. Here was the highest exposure oil, and then these are randomly distributed. You had a control in the middle here, which had no oil added to it. And then you had a far field control over here. And then we went from 1.6 up to 180 liters of oil that was added. So what does that look like? This is one of the limnocorrals once it had been deployed and oil was being pumped onto the water surface. So again, you can see the oil-free zone here. You can see some sampling paraphernalia here, and then the different ports for sampling water and sediment as well. I can't underestimate what an enormous undertaking this was. So to install 9 limnocorrals of 10 meters in diameter, the construction took a team of 12 graduate students more than a month to erect those enclosures. The other thing was, of course, one of the conditions of manipulating the real lakes, again, is that we have to assure Ontario that we can return the lake to its original condition. So we have containment of the oil in these enclosures, but what happens if something would have happened to those enclosures? Nothing did, by the way, but if something had, we had a series of contingencies in place. So there were 4 levels of containment. First of all, the oil was contained in these enclosures. That's 10 meters across, just to give you some idea of scale. Each of those enclosures was then enclosed within another berm that we installed, a series of oil spill booms that were erected by Environment and Climate Change Canada, these spill experts. So that was the secondary level of containment, this boom. Then you'll see some more at the end of the lake. The flow in this lake is in this direction, from the inflow to the outflow here. There's another level of boom there. So even if these were breached and this was breached, we have another level of containment there. That's the 3rd level. There's a small stream at the outflow of this lake, and that small stream flows through a very constricted area here, and we erected what we call an underflow dam. So if there was any oil on the surface, the water encounters this absorptive boom and has to flow underneath, separating the water from the oil. That was the 4th level of containment. And in addition to those 4 levels of containment, we had an extensive monitoring program. So we looked at oil concentrations in water, in sediment, in this lake, and also in the downstream lake. And we never found any, by the way. But that's the sort of contingency plan that we had in place to make sure that we weren't impacting the system beyond what we were trying to do in the enclosures. So once the oil was added, the Boreal study team monitored the enclosures for 80 days. So we left the oil on the surface for 80 days, and we collected samples of air, water, oil, and sediment. And I don't have time to go through all of that, but I'll concentrate on the oil concentrations in the water and the sediments. So recall, one of the things I said was, does oil sink or does it float when it's spilled onto water? So very basic question. The team made daily observations, again, under the tutelage of Jose Rodriguez, and they carefully documented neutrally buoyant tar balls, one of the things you see in the upper image here, and paddies, as well as sunken oil on the sediment of the enclosures. And it's counterintuitive, but the oil that's on the surface of the sediment actually appears as white blobs in this picture on the bottom right. Right, so each one of these is a blob of oil, and the reason it's white is because it starts to be colonized by bacteria and other microorganisms. So what they saw was that the rate of submergence was negatively correlated with the volume of oil that was applied. So the more oil that was applied, the longer it took to sink. That's kind of a good news scenario, because if you're thinking about an oil spill scenario, the more oil that's spilled, that means the more chance you have to remove it from the surface of the water. Smaller spills tend to sink more quickly. And so they actually quantified initial submergence, the time at which they saw the first sinking, and the time at which it was completely sunk, the whole. And again, this is the spill volume here. We go from lower amounts to higher amounts over here. So the first time the oil submerged was around 12 to 14 days for the lower spill volumes, but up to 31 days for the largest volume. So we think that's because the oils tended to cohesively, just hold itself together. This is an important finding, right? Again, it's something that we didn't know much about. Some evidence prior to this had shown that the oil would sink, the Kalamazoo River example, and Lake Wabamun also in 2005 in Alberta. And at the time, we thought that that had to do with aggregation of oil with sediment. But in the ELA lakes, we have very, very little sediment. So the fact that it was volatilized, some of the components volatilized and brought it down to a density that was close enough to water to allow it to sink was an important finding. So what about oil in the water? How do we measure oil in the water, and how much is actually dissolved into the water? Total petroleum hydrocarbons is a really crude measure of how much oil finds its way into the water column. And it generally includes compounds that are, a wide variety in size, anywhere from 9 to 40 carbons, things like BTEX and alkanes and the PACs that I showed you before. So this plot shows concentrations of total petroleum hydrocarbons in each of the 9 limnocorals over time from before the application. So anything to the left of the dotted lines here is before the oil was applied. So we actually monitored these systems before we applied oil. And anything after the dotted line is over time, up to 80 days after that study. And you can see this is the near field control, this is the far field control, and then varying amounts of oil that were added onto the surface of the enclosures. So among the lower oil addition rates, the concentrations of total petroleum hydrocarbons rose pretty quickly, but then they either stayed steady or declined slightly over time. But in the higher volumes, it took longer to get to peak concentrations, but they stayed up there, right? So the more oil you have, the more is contributing to the oil concentrations in the water column as we go along. So that kind of makes some intuitive sense. Now, I've mentioned a couple times this idea of polycyclic aromatic compounds. And again, the simplest one is just this 2-ring structure called naphthalene. But it gets a little bit more complicated than that. Back in 1976, the US EPA listed 16 polycyclic aromatic compounds, or PACs, as priority pollutants because they knew that they were persistent, they were accumulated in animals, and they were toxic. And so these are the 16 PACs that I've shown in the list here. The thing was, those are all the parent compounds. So this is what I would call a parent naphthalene because it doesn't have any branches sticking off of it. We call those alkyl branches, and these are alkylated forms of naphthalene. So you can see there's an alkyl group here, there's a couple of alkyl groups here, there's 3 on this one. The thing about adding alkyl groups is that increases the persistence, the ability of those compounds to bioaccumulate in biota, and also its toxicity. So even though back in 1976, US EPA said these are the most toxic, actually, it's the alkylated forms that we're more concerned with. And I'm just going to show you a little bit of data. Again, this is from Jose Rodriguez's work. These are concentrations of total PAC. So that includes the parent, the unalkylated, and the alkylated. So first of all, let's look at the unalkylated. These are the parents over here. The higher the amount of oil that was added, the more that accumulated in the water column. And here's the controls. I'm going to point out to you here, this is a log scale. So all these major divisions, every time you go up, it's 10 times an increase. Right? So here's the priority PACs that the US EPA told us about. Here, are the alkylated, and here are the totals. These two are basically the same thing. 95% of all the PACs in the water column are alkylated compounds, the ones that accumulate the most, the ones that are the most toxic, and the ones that persist for the longest. But these are the ones that people have been concentrating for a long time on. It's actually these ones that we should pay attention to. And again, the more oil that was added, the higher the concentrations were in those enclosures. So when you talk about polycyclic aromatic compounds, as I mentioned before, they come in various sizes, right? 2, 3, 4, 5 ring. And this is work done by Sawyer Stojanovic, who was a PhD student on the project. And I've just shown 3 enclosures here, and I'll orient you to the axis. These are concentrations in the water column. And again, the study day as you go along the bottom. So the dominant PACs in the large volume enclosures, the ones that had the most oil, were 2, 3, and 4 ring structures. That's really important because those are the ones that are most toxic to aquatic organisms. In the smaller volume oil additions, the ones that only had, 1.5 liters added, yes, there are concentrations like naphthalene there, but they tend to decline over the study period, right? So they're less persistent. Sorry about that. And I also want to point out here that the highest total concentration of PACs was about 2,400 nanograms per liter, which is way below toxicity thresholds for invertebrates and fish. Those tend to be somewhere between 3,000 and up to 60,000 nanograms per liter. So even though we were spilling oil right on the surface of the water here, we're below toxicity thresholds according to the recent data or the available literature that we had at the time. Again, on a mass balance basis, the water column accounts for less than 1% of all the total PACs from the oil. So where did it go? It wasn't in the water column. Where did it go? And again, we found that oil did sink. Well, part of the oil went to the sediments, which makes sense. Again, we know it did sink. Over time, there was a steady increase in the PAC concentrations. I've shown the same enclosures here. To the sediments. And the partitioning or the strength at which those compounds found their way into the sediment had a lot to do with how much they dissolved in octanol, which is a surrogate for biota, for any organism. So the more likely they were to find their way into an organism also was the more likely how they were going to find their way into the sediments. Again, though, these concentrations that we're showing here are well below known toxicity values for the impacts to invertebrates. So those little bugs that live in the sediments. So that's somewhere between 1,400 and 40,000 nanograms per liter. And you can see that that's way more than what we have in the sediments here. Keeps advancing. But I do want to point out that this is data from sediment jars that we put under that oil-free zone in the enclosures. So the oil would have had to find its way onto particles and then settle into those jars. When the Boreal team actually looked at sediments that were in contact with some of those sunken oil blobs, those did exceed toxicity thresholds. So this emphasizes how important it is to remove oil before it reaches the sediments. And that's a really tricky thing to do at an oil spill site. So one of the great things about ecosystem studies is you can measure things in an ecologically relevant setting. So I said PAC concentrations were below toxicity threshold for fish and invertebrates based on what we knew from the literature, but most of that is from single species tests. This is total emergent insects, so things like mosquitoes, black flies, midges, horseflies, caddisflies, dragonflies, beetles. And the way we measure this is using these traps that I've shown in the picture at the upper right-hand side. So these are called emergence traps. They sit on the surface of the water, and any of those insects that emerge from the sediments will fly up into the trap and find their way into a little sample bottle here. And then we're able to collect them and enumerate them throughout the study on a week-to-week basis. So what we've shown here is the total emergence, so the number of organisms that find their way into that jar. And the lighter portions of the bar here are before the spill, and the darker portions are what happened after each of those spills was applied. And again, you have your references here, so no oil applied here, a far field control here, And then as you go across the axis here, increasing amounts of oil. And I think you can see with your eye there that there's a significant relationship. The more oil that was applied, the less emergent insects we would find. And again, this is below any toxicity thresholds from the literature before this time. So this is new information coming out of the ELA that lower volumes of oil will actually cause toxicity in an ecologically relevant setting. So a lot of this data was collected by another graduate student, Tyler Black. He's a very observant graduate student. He has excellent field skills. And he noticed another thing from the enclosures, and that was surface dwelling invertebrates were almost completely absent from the oil-treated enclosures. So water strider, this is one of the surface dwelling organisms, and you've all seen these around Lake of the Woods. They're an important source of food for fish, but they rely on surface tension to be able to stay on top of the water and to remain mobile. So Tyler, based on his observation, and again guided by Jose Rodriguez, performed a series of studies to determine the potential impacts of oil on water strider viability. So the effects were measured in just hours, and it appears that there was a loss of mobility due to the lack of surface tension. So in other words, it wasn't oil toxicity, it was the physical effect of oil reducing the surface tension of the water. And it was related to sheen thickness, how much of the oil was on the surface. So here is the responsiveness of striders. Responsiveness means they actually moved when some stimulus was there. So in this case, in a small mesocosm, if there was a sound that was imparted to the side, they would move. So obviously if you get less responsiveness, they're more moribund, they're closer to death. The darker bars here, anything from going from dark purple to light yellow, indicates an increase in the concentration of oil. And you can see just hours after treatment, you get really almost no responsiveness amongst these oil. Now, to put this in perspective, the oil additions here were very, very small. In fact, there were just a couple of drops in 225 liters, well below any of the thresholds or concentrations that we had even used in boreal. So any amount of oil here is extremely toxic to the water striders from a physical perspective. It reduces their ability to be able to walk on the surface of the water. So just to summarize a little bit from the boreal study, we showed that weathered dilbit sinks. That's a really important finding. We showed that the total petroleum hydrocarbons and the polycyclic aromatic compounds, those oil constituents that are most responsible for toxicity, increase concentrations in the water column with greater spill sizes, but the time to reach those peak concentrations is longer for higher amounts of oil. And we showed impacts to emergent insects well below established toxicity thresholds. And a lot of that had to do with physical effects on the surface tension. So we learned a lot about dilbit, how it behaves in the environment from the boreal study. But back at the beginning of my talk, I mentioned that there was a husky oil spill in the North Saskatchewan River in 2016. And this picture is a picture of the spill response from that spill. And you can see the shoreline degradation here. There's excavation going on. There's soil compaction. A lot of the vegetation is being removed from the shoreline. So all of these can have a devastating impact and longstanding impacts on productivity. And we know for a freshwater system, the riparian area, this area right around the shore, is an important driver of the productivity of that system. And that's why I mentioned at the beginning of the Royal Society of Canada report, thought that it was important to develop strategies that don't affect the shoreline health and stability, but still remove the oil effectively. So that was sort of the basis on which we designed the FOREST project, the Freshwater Oil Spill Remediation Study. So this study was funded by the Canadian Association of Petroleum Producers and the Canadian Energy Pipeline Association. And then that money was matched by an NSERC collaborative research and development grant. And then we attracted additional money from Genome Canada. So it really was, of course, my dog's going to bark when I'm talking. It really was a collaborative effort to study this problem. So the primary objective of the FOREST project was to test minimally invasive remediation methods for removing oil. So We didn't want to go and excavate that material. We didn't want to scrub the shorelines. And we tested 3 different minimally invasive secondary remediation methods. So enhanced monitored natural recovery, I'll talk about each one of these in turn. A shoreline cleaner called Corexit EC9580A. And I won't talk too much about the engineered floating wetlands. Again, that was part of the floater study. When I talk about the efficacy here, I'm referring to limiting exposure to oil constituents in the aquatic environment, and not necessarily to how much of that oil we're able to remove from the system. And there's an important reason for that, and I'll cover that in a little bit. And we also wanted to compare diluted bitumen, the oil that we used for the boreal study, and a conventional heavy crude. And the reason for that is there was a lot of debate as to whether or not Dilbit was quite a bit different from other heavy crude oils. It turns out it's not really. Again, we used enclosures. We can't just release oil into the lake, and we use these as mini ecosystems, except this time we're concentrated on the shoreline environment. So what we're modeling here is if oil spills onto the water and then it secondarily gets deposited onto the shoreline, what's the best way to remove it from that? We know how to remove oil from the surface of the water. It's, there are a number of different instruments to do this, things called skimmers or booms that can remove that oil. So the left-hand side here is a schematic of the shoreline enclosures, and the picture to the right shows actually one set up in our study lake. So again, the yellow part is the flotation collar. Again, it suspends a polypropylene curtain that's sealed to the sediments, and then that goes up onto the shore. Each one of these enclosures was 5 meters wide and 15 meters long, and it encloses about 20 to 30,000 liters of water and the sediments. So they're big enough, again, that we can model the ecosystem, but we can also include things like fish, look at invertebrate communities, and even include frogs as well. So I mentioned that we wanted to compare diluted bitumen and conventional heavy crude. Here's just a graph showing how those different things compare. We measured like 41 different compounds in each one of these. And you can see that, based on the dark bars, which is the conventional heavy crude, and the light bars, which is the diluted bitumen, there's not really a whole lot of difference there. And the reason for that is, remember, diluted bitumen is made up of the bitumen and also the diluent. Well, because we reasoned that oil spilled on the water would take some time before it got to the shore, we weathered that oil before we applied it into the enclosures. So what we did to weather it was to just pour it onto the surface of lake water here in some stainless steel pans, expose it to the atmosphere for 36 hours, collect that oil, and then use that, again, reasoning that it would have flowed across the water, weathered, and then found its way onto the shoreline. Again, I want to emphasize here, 90% of all the constituents that are found, the PACs, the polycyclic aromatic compounds that are found in both of these oils are the alkylated forms and not the parent compounds that the US EPA said way back when were most important. Again, this characterization work was done in Heather Detman's lab at Natural Resources Canada in Devon, Alberta. This is a schematic of how we did this project. So again, an enormous undertaking to put up these enclosures. It took us about a month, a team of about 15 graduate students and scientists. After the oil was weathered for 36 hours, we applied it and we left it on the shore for 4 days without doing a thing. And the reason for that is this is to conservatively estimate spill cleanup response times in remote areas. In a lot of places, pipelines will travel in very remote areas, and if there's ever a spill there, it'll take several days to coordinate a response. We didn't want to model an experiment which was best case scenario in terms of being able to remove the oil. We wanted to model something that was environmentally realistic. So after 4 days, again, we're looking at minimally invasive ways to remove the oil. What we did was to rinse it off the shoreline with a low pressure manifold here. So we're sucking water from inside the enclosure, flushing it over the shoreline, and then we're using absorptive pads here to recover the oil in those enclosures. Each of those floating pads was pre-weighed so that we could determine the mass of oil that we recovered afterwards. And all of that removal occurred over 1 day. So we flushed and we removed over 1 day. And we made no attempt to scrub oil off of any of the vegetation or off of any of the enclosure at all. Again, we want to do something that's minimally invasive, not the sort of aggressive removal that we saw in the North Saskatchewan River. And then we monitored these systems for 400 days after we removed that oil to see what the exposure to oil would be. So after the initial removal of oil, which sort of would be industry standard, if you have an oil spill site at an aquatic facility, one of the things I said is easy to do is remove it off of the surface of the water. So we allowed that to occur, and that was the pads that we were using there. The secondary remediation is, okay, the oil that's left in the system, how are you going to treat it? How are you going to reduce concentrations in the aquatic environment? So we chose 3 different secondary remediation methods. The first is called enhanced monitored natural recovery. This is essentially just adding nutrients into the system to facilitate microbes, bacteria, to be able to degrade the oil in place. They actually use the oil as a form of fuel, but they need other sort of cofactors with it, nitrogen, phosphorus, to be able to use it as fuel. So that's the intent of the enhanced monitored natural recovery. There was some evidence at the time that a shoreline cleaner called Corexit EC9580A would be really effective for being able to coat that oil. It's a coagulant, essentially. So you spray the oil that's on the shoreline, and that allows it to be rinsed into the aquatic environment, again, where it's easy to recover. The issue was that it's not registered for use in Canada. Some of our industry partners thought it could be useful at a spill site. And so because of our special legislation, we were allowed to test this at the ELA. And then again, the engineered floating wetlands is not something I'll talk about today, but if you want to hear a little bit about it, I can talk about that later on. So that's the basic design of the forest study. This is actually the schematic of what we did. So back in 2019, we applied 1.4 kilograms of diluted bitumen onto 6 enclosures in a wetland area and 6 in a rock and cobble area. Why the replicated study design here? We know that bacterial communities are quite different in a rocky cobble environment than in a wetland environment where there's more organic, like peat type of sediments. However, it turns out when you put it up an enclosure and you try and unseal it to the bottom when there's a rock and cobble enclosure or rock and cobble substrate like this, it's very difficult to be able to seal them. And so we won't talk too much about the rock and cobble because the data are very messy from that. So we had in 2019, that leaves us with 2 reference enclosures, no oil added, 3 that were treated with diluted bitumen and then enhanced monitored natural recovery, and then 3 that were treated with diluted bitumen and then treated with shoreline cleaner. And remember, we wanted to compare diluted bitumen with conventional heavy crude. So in 2021, we replicated that study design. This time we had 3 reference enclosures, 3 that were treated with conventional heavy crude, and then enhanced monitored natural recovery. What happened to the shoreline cleaner? Well, it turns out the shoreline cleaner wasn't very effective for helping us to remove the oil. And it was also pretty toxic to the aquatic invertebrates. And so that's one of the things that's come out of our research is that the industry is no longer pushing for the use of this shoreline cleaner. They've, sort of abandoned that. And in fact, the manufacturer of that product no longer manufactures it. So I'll concentrate on the enhanced monitored natural recovery comparisons for the 2 studies. Now, I didn't talk too much about how much we were able to remove, but here's the overall statistics. Only 4.5% of the oil was removed in 2019. 23% was removed in 2021. So you'll hear a lot of talk about at oil spill sites that 80 to 90% of the product has been removed. Those are sometimes very difficult statistics to believe. And in fact, the actual removal rates are probably between 20 and 40% if you're being optimistic. So we assessed the effects of the oil exposure in 6 different themes. And I don't have time to talk about all of those again today. I mean, we looked at microbes, the bacteria, we looked at the invertebrates, we looked at fish and frogs, and we even looked at whether or not the oil was toxic to the vegetation on the shoreline. But again, we're interested in the chemistry, the polycyclic aromatic compounds, and specifically those 3 to 4 ring structures. So that's really where we placed our emphasis on this study. So we looked at PAC concentrations for over a year in the enclosures, and that's the reason why you see these shaded portions of each graph. That's the winter. We were actually sampling during the winter as well there. Partly because it's believed that a lot of the degradation of oil over the winter is relatively minimal, but it actually does occur to a greater extent than we originally thought. So all of this chemistry work was done by Lisa Peters, who is now, again, a research scientist at the ELA. At the time, she was working in Greg Tomey's lab at the University of Manitoba's chemistry department in the Center for Oil and Gas Research and Development. So a couple of things to note here. I haven't shown the breakdown by ring structure here, but these are all predominantly 2 to 4 ring PACs, the ones that are most toxic again. Peak concentrations in the Dilbit study were around 9,800 nanograms per liter. And that occurred within about 6 days after the exposure. But the shoreline washing agent, again, here maintained concentrations for longer. Another one of the reasons why we didn't, continue with it in 2021. Peak concentrations were almost 7 times lower for the conventional heavy crude, so around 1,400 nanograms per liter, and also appeared earlier on day 3. But the important thing to notice from each one of these graphs is relatively quickly after the spill, so here we're looking at sort of 50 to 70 days, you get down to near baseline concentrations of PACs in the water column. So how does that fit within the context of what I showed earlier for the boreal studies? So here's 2 plots of total PACs from the 2 boreal study enclosures that were nearest to our oil to water ratios. So in that study, Sawyer observed that the time to achieve peak concentrations was longer for larger oil to water ratios. So peak concentrations were around day 2. And for us, we were again around day 3 to 6. Peak concentrations in our study were also much higher than what they saw in the boreal study. So they peaked out around 700 nanograms per liter. And I told you we just peaked out around 7,000 nanograms per liter and even higher for the Dilbit. And the reason for that, we think, is it's the oil that's deposited on the shoreline that's continually being mixed by wave action. So that's a really important difference. So some of the lessons that we learned from the boreal study didn't necessarily apply to the forest study. And again, really important to know that the minimally invasive methods that we use to respond to these oil spills resulted in relatively low concentrations in a relatively quick manner. So these are depuration curves. So the time to reach 50% concentrations using enhanced monitored natural recovery in 2019, just 7 days after the oil was exposed, we're down to 50% concentrations. In 2021, it's around 8.5 days. So relatively similar to what we saw with diluted bitumen. But for the shoreline washing agent, again, this is something that helps to coagulate the oil so you can rinse it into the environment. It's longer. It takes up to 12 days. And again, we had some evidence that it was pretty toxic. So once we noticed that PACs, we noticed from the boreal study that they tend to sink and that PACs can be found in the sediments. We also looked for PACs in the sediment of the forest study, and we found relatively high concentrations of PACs in the sediments, but a lot of it was made up by this compound here called ratine, which is a 3-ring PAC. The thing is that ratine doesn't come from oil. It comes from forest fires. And so about 75% of all the PACs in the sediments came from ratine. It's forest fire activity, or it's a forest fire signature. So when we corrected for that, and then compared to baseline enclosures, we found that there were relatively low concentrations of PACs in the sediments. So it wasn't coming from the shoreline into the water and then back into the sediments. So we did actually revisit this and extensively sample the sediments to see if there's heterogeneity in that. In other words, are there the blobs that we saw in the forest or in the boreal study also found in the forest study? Now I wanted to touch a little bit on effects. So in other oil-related studies that we performed, emergent insects, like I showed you for the boreal study, are amongst the most sensitive to the presence of oil. And again, that might be a physical effect. So again, we use those emergent insect traps in the 2019 study and in the 2021 study. In 2019, well, in both years, actually, with both oils, we didn't see any significant effect on the species richness, which means the variety of different kinds of organisms that you see emerging from the sediments. And there was no effect of on total abundance in 2019, but there was an effect in 2021. So in other words, these bars here showed less emergence, and these are the oil-treated enclosures, than these bars here, which are the reference enclosures. So again, one of the things we wanted to compare here was Dilbit versus conventional heavy crude. The recovery of oil was quite similar. This is a little higher than that figure that I showed you earlier, but this is an adjusted. So around 7.5% to 24%. So a lot of the oil remained in the system. In fact, this is the residual oil that we were studying. So around 1,200 grams in the diluted bitumen and around 1,100 grams in the conventional heavy crude. Peak concentrations around between day 3 and day 6, and return to baseline around day 60 to 70. We didn't find PACs in the sediments in either case. The effect on the microbial community, in other words, how did the microbial community respond to the presence of oil? Did we see more microbes that were capable of degrading that oil? Not really. It's a very difficult thing to get a handle on that, and the community changes quite rapidly. We do know that there are microbes, bacteria in the system that are quite capable of consuming that oil, and that's the reason for why we found that a lot of that oil was removed from the system. And again, the effect on emergent insects, no effects in the 2019 study, but we did find lower abundance in the 2021, which is more consistent with what we found with the boreal study. So just to finish up here, one of the remaining questions after the forest study was completed was, okay, we found low concentrations after 60 to 70 days in the water column. We didn't find it in the sediments. Where did the oil go? And was it still sort of contained within this region where the oil was deposited? Because we deposited the oil in the water and it found its way onto the shoreline. Was it still there? Is it still undegraded? And so we were interested in whether, the actual physical removal process that's usually used at an oil spill site would mobilize some of that oil or recover some of that oil. So Enbridge funded a follow-up study that we called the Prove It study, the physical removal of oil to validate in situ treatments. So in the spring of 2023, we removed 25 centimeters of soil and vegetation. So the zone that was potentially containing that residual oil using our shoreline cleanup and assessment technique evaluation. So these are the enclosures from 2021. So there's 2 reference enclosures and 3 that were treated with conventional heavy crude. And we excavated all of these. We even excavated 1 reference enclosure because we wanted to see what the effect of just excavation would have on the near shoreline environment. So this is a view of one of our enclosures before excavation, and then from the same vantage point after excavation. So 25 centimeters, that's a lot more than 25 centimeters. What I'm talking about is vertical 25 centimeters. That's the maximum height that a wave would have reached up onto the shoreline. And this is an overhead drone shot showing the extent of material removed from enclosures 1, 3, 4, and 5. And this enclosure was left intact, a reference enclosure. So that amounts, all of that material was put into bins, and then we took that off-site, dried it, and homogenized it so that we could look at concentrations of oil in it. That amounted to about 900 kilograms of material that we removed from that shoreline. So first we wanted to know whether or not the excavation itself liberated some oil that had been on the shoreline. So what I've shown here are the concentrations of the various types of PACs, 2, 3, 4, and 5 rings before excavation on the left. So you can see there was oil, some residual oil, very low concentrations here before the excavation. And we did mobilize some, but it very quickly returns to near baseline conditions. So we really weren't excavating or removing any of the oil. We didn't mobilize any PACs. We wondered whether or not if we did mobilize it, it was perhaps bound to sediment that was mixed into the water column. And so we looked at turbidity, and it was elevated in all the excavated enclosures, especially enclosure 1, but it returned to reference levels by day 32. Some of those enclosures exhibited elevated total suspended solids throughout the season, and specifically enclosures 1 and 5. But when we took that material, it didn't have any significant PACs associated with it. So the table shows concentrations of total PACs in the soil and the vegetation that we removed from the shorelines of each of those enclosures in the red box here. So if you accept that about 1% of the oil is accounted for by PACs, then the amount of oil we actually removed from the shoreline was only about 20 to 30 grams. Remember, there was about 1,100 to 1,200 grams of residual oil that was left. So there was very little oil that was left in the shoreline soil or the sediment or the water column or the vegetation materials. So when we break those PACs down into groupings, there doesn't appear to be any significant elevated concentrations in any of the enclosures, and specifically not the 3 to 4 ring PACs that we should be concerned about, because those are the ones that impart toxicity. So just to generally conclude then, very rapid declines of PACs in the aquatic environment adjacent to the oil spilled onto the shoreline. There was very minimal oil that was deposited to the sediments and none to the shoreline and soil and vegetation. So the general conclusion then is that these minimally invasive ways to respond to oil spills in a shoreline environment can be a very efficient way to respond to it without damaging that shoreline. In other words, you can rely on the bacterial community to degrade some of that oil. And then these are just some of the other general conclusions that I'll let you read. And I'll stop there and hope that there's some questions. And yeah, I'll stop there. Thank you.
Yeah, Vince, Brady has 2 questions. If he could unmute, please.
Yeah, couple of the questions that I asked. What is the real name of the boreal lake, Vince? Or was that the name that you gave it? Is that, or it didn't have a name, or?
No, so all of our lakes at the Experimental Lakes Area have these very romantic names, and that one is called Lake 260.
Okay.
So all of our oil studies have occurred in Lake 260.
Okay. The other question I had was the white oil that sank to the bottom. Was that dredged when the testing was done?
Yes. So, yeah. So the oil itself wasn't white. It's the biofilm, what we call biofilm, the algae and the microbes and the fungi that colonize it. They sort of look white. Yeah. At the conclusion of that study, inside the enclosures, I mentioned before, Environment Climate Change Canada was a huge partner in that project. Bruce Hollowbone was the scientist who was collaborating with us. So he and his team came and vacuum removed that sediment from the bottom of those enclosures before those enclosures were dismantled.
How long, if it had been left, would it have been before it completely was eaten up by the bugs or whatever that the microbes? How long would it sit there before that was cleaned up?
Yeah, so the results from the boreal study are quite a bit different from the forest study. And the results from the boreal study suggest that there was minimal uptake of the carbon from the oil into the microbial community. So, and evidence from other oil spill sites says that it would take decades for that oil to be removed. So it's very slow because the complexity of those big PAC molecules that I showed, it's very difficult for microbes to be able to break them down. One of the limiting steps is each one of those rings has to be broken open. So the reason the long chain alkanes are broken down so easily is because microbes can break them into 2 and 3 carbon fragments that they can use to generate energy But before they can do that, they have to break those rings. And when you have sort of something that looks like chicken wire, it takes an awfully long time to do that. We don't know whether or not the process in the forest study was accelerated because of the addition of the nutrients, or because the microbial community is different in the shoreline environment, or because of the mixing energy that would have been imparted by the waves in that shallow area, and especially because of the availability of oxygen in that area.
Okay, like I would imagine that like the oil spill, the Valdez, and the oil spill in the Gulf, um, from different climates, different temperatures, um, would be totally different from each other. Am I correct in that?
Yeah, absolutely. So, actually, it's a really interesting comparison, Exxon Valdez versus the Deepwater Horizon. So, Exxon Valdez still has areas where you can find significant concentrations of oil. Deepwater Horizon, there is that in some of the shoreline environments. In some of the deeper parts of the ocean, very difficult to find those deposits of oil. And I mean, the ocean is an enormous, vast area to look in, but it looks like in the ocean, there always is natural seeps of oil. And so the bacterial community there is primed to be able to remove those types of constituents. And that may have something to do with it as well. But certainly in colder water environments like Valdez and in other oil spill areas, there has been very slow degradation of the oil.
Great questions, Brady. Is that it, or do you have one quick more before we move on to the next question?
Well, I just wanted to tell Vince this is a great presentation, and thank you very much for presenting to us. So yeah, we can
Hey, you're
move.
quite welcome. Thank you. Appreciate it.
Vince, Chris Hay has a question for you.
Yeah, it sounds like you made some important new discoveries about dilbit sinking and invertebrate impacts and so on. So what was the impacts on all your findings on actual oil practices, whether development, transport, or probably mostly remediation? So like hopefully your findings have helped change things for the better in terms of how things are done. So has there been any changes so far, or how does that look, or how do you think that'll go going forward?
Yeah, well, I think there's been, I mean, it's not just, not to attribute all of that just to us either. There's been an expansion of the, after the Royal Society report noted that there was very little that was known about how oil behaves in fresh water. There's been an increase in the research in that field for sure. I mentioned before the use of the shoreline cleaner is not something that's, being considered in freshwater. We sort of feel like that's a major impact, or at least a small portion that we can contribute. We showed that it's especially important to be able to get to an oil spill early before it sinks. And I think that's probably having some impact on the availability of spill response equipment and the importance that's being placed on mobilizing. So we've seen some, and again, it's the totality of the information that probably affects this, but we've seen some moves to harmonize how oil spill responses are coordinated, in recognition that, time is of the essence to be able to get that oil off the water.
Great. Thanks, Vince. Willatuvi has a question and is unmuted.
Hello. Thank you for your presentation. It was very informative, and I am by no stretch, I am not a scientist. I'm only a cottager. We have a cottage association, and we are wondering about the use of what's called RAP, or recycled asphalt, on roads that are close to the water. And we have no idea whether that's a good idea or whether we should be using just crushed rock. But yet the government uses asphalt on highways everywhere, and they're right beside lakes. And I understand that, your study looked at oil spills, which are, a lot more oil, but over a period of time, like we're talking decades, and the Trans-Canada Highway, I mean, we're talking 100 years. Does that does that have an effect, or are we making a mountain out of a molehill?
It's a good question. Thank you very much. So I mentioned before that diluted bitumen is made up of, thousands of different compounds, and some of those large PACs that I showed that look like chicken wire, one of the categories of that is called resins, and the other category of that is called asphaltenes. And asphaltenes have an enormous, large, multi-ring structures of PACs. Again, they're very difficult to break down for microbes. It takes decades, multiple decades for them to be able to break down. But those are the very same constituents that, are present in bitumen that would also be present in the asphalt. So I guess there's a reliance on the fact that they're very difficult to break down and that there won't be any weathering. The reality is that there's probably some concentrations of PACs that are being liberated from that asphalt, but in a very, very slow manner, such that the concentrations that would be found, during a heavy rainstorm that would wash into a nearby lake would be negligible. That's the assumption anyway.
Okay, thank you.
You're welcome.
I don't see any other questions. If anyone has a quick question, stick your hand in the air, please. Otherwise, I'll hand it over to Megan.
Well, I don't see any hands and want to acknowledge that we're a bit over our time for the timeline. So just let me know if you need to jump off, Vince. But mostly I want to say thank you so much for everyone tuning in. And thank you, Vince, for sharing this presentation with us. That was fascinating. I know I learned a lot about the complexity of these experiments and the incredible amount of work that's gone into making them possible. I also want to acknowledge and thank you for that thoughtful approach that you said right at the beginning of your presentation, that a lot of these experiments take so long to even get off the ground because you're working with the communities before even beginning the research, which I think is such an important part of doing the type of work that we're doing and doing it well. Yeah, I guess Vince has his email there, so if there are any final questions, I'm sure you can email and reach out to him. But otherwise, Vince, thank you again, and thanks for everyone who's joined us in this session. Recording of today's webinar will be available sometime soon on our website at lowwsf.com/askanexpert. Feel free to revisit it and share it with folks. We'll also have some other webinars coming up soon. The next one is on October 28th with Dr. Chelsea Rockman from the University of Toronto, talking about microplastics and freshwater health from a plastic project at ELA as well. So thank you, everyone. Take care and enjoy the rest of your Thursday.
Thank you, Megan, for bringing this to us.
Yeah, thanks, Brady.
Make sure you watch Chelsea's talk. It'll be great. I've seen her speak before, and the work she's doing is really very cool. And she also extends it into the Great Lakes. So I think you're going to be entertained.
Amazing. Okay, thanks all.
Bye-bye.
Bye now.